Hi, you're watching DNA Today, a multi-award-winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor, and your host. We're diving into the cutting-edge world of variant curation through deep mutational scanning and multiplexed assays of variant effect. Now, if you don't know what any of that means, no worries. To break down what it means, I'm joined by leading experts, Drs. Starita and Fowler. Dr. Leah Starita is an associate professor at the University of Washington and co-director of the Brotman Bay Advanced Technology Lab. And Dr. Douglas Fowler is a professor of genome sciences and co-director of the Center for Multiplexed Assessment of Phenotype. So we're going to explore how these groundbreaking technologies are transforming our understanding of genetic variants and paving the way for precision medicine, because a big part of variant curation and actually having a variant curated of not being a variant of unknown significance, I think, is achieving that precision medicine of actually being able to do something with that. Well, Leah and Doug, is that how you would explain variant curation, of figuring out, okay, this variant in a human genome, because we're just focusing on human genetics for this, is either it's pathogenic, it's going to cause an issue in that person's body and have symptoms from it, or it's benign, it's just part of human diversity, it's beautiful, it's great, there's no issues with it. Anything that you would add to that before we get into deeper of these different technologies of how to variant curate?
I mean, I would say that is the simplistic view into which we break things down now. Either things cause disease or don't cause disease, whereas, there is a spectrum. But I think for potentially this podcast, it might be easiest to just kind of think of it that way. There are a lot of variants that will very high likelihood to cause disease. These are your Mendelian conditions. And then there's variants that are very low likelihood to cause disease, or we're going to call those benign, or, just kind of passenger mutations if you were in the cancer.
Yeah, I would add just a couple of observations there. One thing to know is that, like, you might naively assume that most variants that we've seen in human beings are classified as either pathogenic or benign, but you would be wrong. About only about 10% of the variants, at least in the ClinVar database, kind of where we collect these things, are classified in that more definitive way. And most of the variants that are in there are actually called variants of uncertain significance. And maybe this kind of underlines why what we're going to talk about is so important, right? But the bottom line is that often when we, look in a genome or try to interpret the results of a genetic test, we can't actually figure out what the variant does. And so that statistic kind of sticks with me when I'm thinking about, like, why I get up in the morning. So anyway, that's what I would add to Leah's excellent answer.
Wow. So around 10% of human variants are classified to not be in that in-between. It's one or the other. Leah, can you see if I got that right?
It's not an in-between. It's we have no idea. So in-between would mean maybe this variant might cause disease and 50% of the, well, doesn't even really mean that. But it's not an intermediate. It's we have no idea. So what's happening is that people are getting genetic testing. They're finding these single nucleotide variants or other sorts of variants. Just one letter change in these really important genes that we've known a heck of a lot about for 20 years. And we still are just like, that's a new one. Weird. It's really, it can be very difficult to predict whether a single nucleotide variant that changes an amino acid is going to blow up the protein, or it's just going to sit there and it's fine.
And there's a big difference between, all right, if it just sits there and is fine, or it blows that up and that protein is not going to do what it's supposed to. So knowing how challenging this is, and for those that might be listening later, we're recording this in January 2025, because I think that's going to be very, important, just knowing timeline of, like, when we're having this conversation. So knowing that curating variants is so difficult, can we dive into then what deep mutational scanning is and multiplex assays of variant effect? And talking about what are these technologies and how is it helping towards that goal of having more variants classified?
Doug, you invented the method. You should do it.
Yeah, Doug. Yeah, that's your question then.
Happy to I'm happy to answer that question. I think that there's one thing to maybe a step zero that we should talk about, which is when we want to classify a variant that we see in a clinical test, a genetic test, let's say. We have to assemble, or clinicians assemble, or variant review scientists assemble different types of evidence. And so one of the types of evidence that you can use is what we call functional data. And we'll probably dig into exactly what functional data means later. But what it really reduces to in plain language is doing an experiment of some kind that allows you to know, at least in that experiment, what the variant does relative to a reference sequence or like the wild type. And so the whole idea of mutational scanning is basically to take that type of measurement, which in the past was done one at a time. So you see a variant you're interested in, you go to the lab, you make a cell line, you do an experiment on that cell line, you say, this variant causes the cells to stop growing, let's say. And so now I know something about that variant. The whole idea of a mutational scan is to take that one at a time process and parallelize it so that you can, instead of measuring the effect of just one variant in your experiment, you can instead measure the effect of thousands or tens of thousands of variants in one experiment. And that number 10,000 is kind of a magic number because it's about 10,000 variants that you need if you want to test the effect of every possible single letter change in like your typical human coding region of a gene. And so we call experiments that are focused on amino acid changes deep mutational scans like that. And then MAVEs, multiplexed assays of variant effect, are actually it's a parent term. So that's all the experiments that you can do with variants that are parallelized in the way that I told you. So that would include like non-coding regions, introns, all the stuff that's not, coding changes. So that's what those terms mean in kind of a very high level general sense. But if you remember something, it would be like, these are a way to parallelize experiments that tell us about what variants do.
And that's so much more efficient of testing. You said like 10,000 versus one. I mean, that's monumental of just how fast we can get more data to then be able to classify variants and have that information. Because as you said, it's not even just variants that are occurring in the exons, that we're looking at the coding part of the genome. So that 1% of the genome that we kind of consider active, it's also, we need to look at the 99% of the genome that's not necessarily active, because that could affect the other parts that actually end up being expressed or not. I think that's one of many reasons why this is so complex, and that we're not as far as I think people realize. Our Human Genome Project finished in 2003, so we're like 22 years out from that. And yet some people say, wow, we've come so far. And then on the other end, it's like, wow, we really haven't gotten that far in terms of understanding all of these variants. So I don't know. People have different opinions that have expressed it on the show, but I think that's just interesting to see it that way. And it's just what a great method to be able to get all of this data at once. Leah, did you have any other thoughts on just MAVEs and how this process is actively helping us now?
Yeah. So one of the things I wanted to add was that it's that same revolution in DNA sequencing that has allowed these MAVEs to happen, right? So the same revolution that has kind of exploded this VUS problem, right? So for genomic sequencing, clinical sequencing, whatever you want to call it, full genome sequencing versus genetic testing, that has really exploded because sequencing has just gotten a hell of a lot cheaper, right? So, genetic testing has gotten cheaper, but our ability to actually understand, so we can read genomes, we can read exomes, we can read, small gene panels, but our ability to actually understand what we're reading has not evolved as quickly. So we're kind of winning. We're winning on one side, but we've got this major barrier on the other. And it's that same technology, the DNA sequencing, that has enabled these parallelizable assays that Doug just told you about. So kind of the two fields are kind of evolving almost at the same time, which I think is really kind of cool. So it's that sequencing that is that enabling technology for both.
We're really good at reading genetics, but we're not necessarily good at understanding it in terms of like, okay, we can read this gene, but do we even know what this means and how that affects how the protein is going to function or not function, or somewhere in between, if it's less of a function, all the nuances there. And when I think about, as someone that has never done variant curation, I think that's important to say. I know you both know that, but for those in the audience that may not know me as well, for someone that hasn't done this, I think about, all right, if we're looking to see what the effect of a variant is, let's check out a mouse model. Let's check out, as Doug was explaining earlier, let's have cells that have that variant that we're looking to understand more about. How does that affect the cells? Are they growing? Are they not growing? Is something different? Also looking at population frequency. Well, have we never really seen this variant, or we very often see it? And we're like, oh, well, if it's in a lot of people and all these people seem to be relatively fine and not really have symptoms, that's probably okay. So those are the aspects I think about when curating a variant. So how does MAVEs then play a role in this? Is it kind of like if we have a spreadsheet of all of these different, I'm such a spreadsheet person, right? I'm a type A person. So if we have all these different factors, like, okay, population frequency, how do the cells look, mouse model, all these different things, is the information we can get from MAVEs kind of another piece to the puzzle, or is it potentially replacing some of that, or more important than those aspects?
So some of the strongest evidence that we can have is actually this population frequency that you just talked about, right? So just as you explained, if a variant occurs way too often, like more often than the disease itself, right, it cannot be pathogenic. So that's like standalone
just straight up statistics.
Right? Okay. There's a big problem with that, is that, you know, these genetic studies are mostly white Europeans.
Yeah.
Okay, so they're fairly biased, and also the people who have had access to genetic testing, generally white Europeans. Okay, so the variants that are in ClinVar, the variants that are in these big genetic studies that give us these allele frequencies globally are not so global, and so therefore, you know, when who is accumulating variants of uncertain significance in even at the University of Washington. The people of non-European descent tend to have more VUS in their genetic test results than white Europeans. So that kind of leaves a footprint then on the field of genomic medicine, where, you know, people from non-European ancestries are now shouldering the burden of this VUS, and therefore genetic medicine really is not working for them. And so that's, you know, that's kind of a, you know, these decisions to who's included and who isn't coming from up high kind of affects how medicine is practiced on the ground. And so that's a bummer. And then, Doug, do you want to talk about the scale, mouse models versus MAVEs?
Yeah, I mean, I think that's the other thing to mention here. So we talked about population evidence. There are a few other lines of evidence that many other lines of evidence that go into a variant interpretation, and they can kind of broadly be grouped into ones that are more scalable and ones that are less scalable. Population frequency is a great example. Like we've sequenced millions of people now, or genotyped at least, and we kind of know what the really accepting populations we haven't looked at least in white Europeans. As Leah said, we know what the common variants are, but most of the variants we find when we sequence genomes are rare, like ultra rare. So that's an example of like a non-scalable type of evidence. There are other ones like that. We really like to know about the segregation of a variant in a family that's affected, right? And we can, by knowing who gets the disease and who doesn't, and who has the variant, who doesn't, we can generate evidence that the variant is pathogenic or benign. But that's hard evidence to scale, like going and getting people to test their family members. And that's a hard conversation. Like there's a sociology problem, money, the other hard problem, you know. So then we're left with like, what kind of evidence can we scale? Could we feasibly get for every variant in the genome? And functional evidence is one of those kinds of evidence from a MAVE, right? So you might say, well, we can collect functional evidence with mouse models, and you can, and that's great. But mouse models are expensive. Mice take up a lot of space. So you can't do 10,000 mouse models, right? That would be a Herculean effort, and that would only get you one gene. And so the magic of mutational scans is that, and we've, written about this, and it is our mission. Me and Leah, have worked many years towards this goal, and I think we'll achieve it eventually, is to, along with a whole bunch of other people in our community, create variant effect measurements in the lab using these technologies for every variant in every disease-related gene in the genome so that we have at least some evidence for every variant when it shows up in the clinic. And, that's kind of what we're trying to do. I think, to circle all the way back to where you started, do I think that variant effect data or functional data will be somehow more important than these other data types? I think the answer is no, right? I mean, the clinical community has done a really fantastic job of articulating rules for interpreting genetic variants. And, those rules tell you how to combine the different types of evidence. But so in that sense, I don't think it will end up being like more or less important than other types of evidence. But one thing that we hope to make true is that it's available. Like, so the evidence that's available is important, right? And we hope that variant effect data, it is already available for a number of genes. And our view is that it should be available and could be available for every gene.
Yeah. And
it doesn't replace mouse models either, right? Like there's a place for, holistically understanding the effect on every organ system in a mammal, right? Whereas, the part of the key of these high throughput assays is we're measuring kind of simplified phenotypes to gain that scale, right? So that it's a scalable method that gets you some information, which is better than no information, which is what we're the current paradigm, right? So, it's not a replacement. It's a different beast altogether.
Yeah, it's another tool that we really need, but it's, from what you both are saying, it doesn't sound like it's going to replace what we currently have, but it's adding to, here's even more information, and especially saying we can't have a mouse model for every single variant. I mean, that's just not feasible. As Doug said, that'd be a, huge project. We don't need that either. But as Leah was saying, like, for certain conditions, especially if it's like, a lot of rare diseases affect kids. So, all kiddos with this certain variant, the parents come together, they have an organization, a nonprofit, and they say, we want a mouse model for our kids. They all have the same variant. We want to see how it's affecting them. Maybe there's a ASO or something in the works, antisense oligonucleotide. Now I'm getting it wrong.
Yeah.
Yeah. Okay. I should know that. And Lorem, I work for them. I got to know that. Anyway, so there's a time and place for it, but not all the time. And that's going to be way more expensive than using MAVEs. Something I wanted to ask you about, Doug, is as I was doing a little prep for this episode, I either came across or you or one of your colleagues sent me a paper that you're a co-author on. It was published at the end of 2023, so a little over a year ago from when we're recording. And the paper is, Will Variants of Uncertain Significance Still Exist in 2023? You knew I was going to ask you this. Or not 2023, 2030. Yeah. Now we're 5 years out from that. So it's been a little bit since you wrote the paper. So it's the name of your paper, but that's the question I have. Will it still exist 5 years from now? Because 5 years seems very soon. It's been documented on the show. I've said before, like, I'm sure by the end of my career, I'm 29 for reference. Once I have grandkids, maybe we'll be at the point VUSs aren't a thing. I'm going to sound outdated referencing VUSs. But you're talking about maybe 5 years from now, VUS isn't a thing. To me, that seems like, that's like, how could that happen so fast? But you would know better than I would.
Well, I don't know. I mean, you're a genetic counselor, so I'm not sure that I know better than you, but okay, look, the paper you're talking about is a bit of a hot take. I wrote it with Heidi Ream, who's a, leader, one of the key leaders, actually, I would say, in clinical genetics. And, the thesis of the paper is that, I mean, obviously, we don't want to argue that every VUS is going to be gone. I think there are lots of variants that will still have trouble figuring out what to do with. But the idea of the paper is that there's sort of a confluence of technologies that allow us to imagine a world where VUS is not like the primary challenge that it is now. As we sort of laid out in this discussion so far, it really is kind of the key blocker for precision genetic medicine at this point. And, I think we've demonstrated in papers that we being me and Leah have published that just adding this functional evidence from MAVEs can get rid of lots of the existing VUS, like somewhere between 20 and 70%, depending on the gene. And we already have that kind of data for 40 or 50 genes, and we and others around the globe are generating it at a pretty brisk clip. The other big thing that's happening is that computational variant effect predictors, so AI models that can predict what variants do, that's another line of evidence. And some big changes have happened in how the clinical genetics community uses those tools. And so
and how good they are now. They've got much better.
And how good they are. And so, those changes are also going to help provide more evidence. And so I think I will stick by my prediction that we will, be much less focused on VUS in 2030 than we are now as like the key problem. And I think some of the, you mentioned ASOs, antisense oligonucleotides.
Thank you.
As like an example of that's a variant guided therapy, right? So that's a therapy that only works for some variants. And if I had to place a bet, I'd be betting that the grants that I'd be writing about and the like hot takes I'd be putting in the literature, in 2030 are going to be focused more on, okay, now that we've articulated a framework for understanding variants, and maybe we haven't gotten to all of them, but at least we know how to do it now, and it's just process to get there. We're now talking about how do we articulate a framework to figure out how to develop a treatment for a variant. Not every variant, again, there'll be a long tail, right? But how do we actually take action on the basis of knowing, okay, well, this is the pathogenic variant. What do we do next? And so anyway, I stand by the sort of thesis of the paper as we wrote it. But I agree, 5 years is coming fast. And so Leah and I are hard at work. And hopefully we won't have another conversation in 5 years where you say, Doug, where did it all go wrong?
Or, wow, you did such great work. VUS isn't even mentioned anywhere. There's not really a lot of presentations about it anymore. Like, it's not a focus. That's the hope, right? Like, we don't want to be talking about VUSs because we want to just have answers with it. And so something else I was wondering about is with MAVEs, I had not heard of this concept until I connected with you and other colleagues of yours. How many other people, institutions, labs are doing this? That might seem like kind of a simple question, but is it you guys are kind of the headquarters for this and this is where it's happening? Like, how many other places are doing this? In order for us to get to not having a lot of VUSs and having MAVEs be a big part of this, it can't just be you guys, unless we were able to clone you guys and, all kinds of other cool genetic stuff to get there.
Yeah. So there's a couple of things. We have Doug is the co-leader of a consortium alliance for Atlas of Variant Effects Alliance. So we kind of co-founded it's kind of a grassroots community around this field, right? So we're trying to be able to encourage more people to get in the field. We're trying to encourage the people who are generating the data and and using the data. So the geneticists and then the molecular biologists to talk to each other and figure out, what each other needs. We're trying to write some rules and standardize how people are communicating about this data. And so to really kind of be able to generate like the infrastructure needed to grow the field. And there's an annual meeting coming up in Barcelona, which is not a terrible place to be.
Put a link in the show notes for people that want to go or in the area.
It's called the Mutational Scanning Symposium. I think the registration should open pretty soon. It's January 2025. And I think the meeting's May 21st to 23rd in Barcelona. And this meeting is great. We hear a lot of unpublished work from people across the field. This one will be heavily, because of the people who are organizing it, focused on a lot of the AI methods and the intersection between MAVEs and AI, which is pretty cool. So there's that community. And then there is a National Human Genome Research Institute, part of the NIH consortium called the Impact of Genetic Variation on Function. And there's funding there for a bunch of labs who are systematically perturbing the genome and measuring, linking genotype to phenotype. What am I missing? The Sanger has a big push as well.
The Sanger Institute?
Yeah.
Yeah.
Of course, got a lot of firepower over there. What else am I missing, Doug?
No, I think you got it. I mean, the stat you might want to know is that the alliance that Leah mentioned has about 600 people in it. So, I think we've achieved critical mass. Like, I think now there are enough people working on this problem and data is appearing fast enough that it's not unreasonable to think that we'll get to not every last gene, but, a majority of the genes where clinical geneticists would really like to have this kind of data and it is practical to generate it. Because Titan, as Leah is famous for saying, will be done by somebody else, not our labs. But for those genes that are practical and where there's clinical need, I think, we have a community now that's focused on making the data and making sense of the data and working with clinicians to translate the data.
And how easy is it to actually do this process with the MAVEs? I mean, is it like something I think about that is there's really hard ways of genetic editing, say like TALEN versus CRISPR kind of has, I haven't used it. If there's anyone out there that wants to bring me in their lab and show how CRISPR is used, I would absolutely love that little field trip.
On over.
But sweet. All right. I'll be in Seattle in November. Well, how easy is MAVEs? Is it like you have to really know what you're doing, or is it like, more intermediate? Like it wouldn't be too hard for labs to say, we should start using this process and technology because we're focused on a certain gene? Or is it more these people in your community of researchers that's like, all right, yep, nope, it's much more involved?
I think, one thing that we haven't talked about that's worth appreciating is that, like I said, MAVEs, that's a parent term, right, for all these different kinds of assays. And they've been elaborated by this amazing community I was telling you about. And there are ones that really span the spectrum from what I would consider to be fairly easy to implement. Like, if we're going to use a skiing metaphor, they might be like blues.
Okay.
So like your double black diamonds to your like, I'm going to ski off the top of Everest, And I think there are some types of these assays, and you're beginning to see. So I mentioned this alliance. It's got 600 people in it. I would say that is now. I don't think that we have the majority of people using this technology out there in the world in the alliance, because the alliance is pretty focused on solving this clinical problem that I told you about, and lots of other people want to know about what mutations do in proteins or viruses or whatever. And so they use these technologies from that perspective, but they don't care. They're not interested in the clinical problem we talked about. And so I think the technology is matured to the point where it's like, you have to know how to clone stuff. You have to know how to do library scale stuff. So it's much harder than, I don't know, doing a mini prep, but it's no longer, I would say, a specialist method that requires years and years and years of specialized, knowledge to really make work. So it's kind of, and then there are technologies like we're developing in my lab and Leah's lab that are the next thing we want to be able to measure at scale. And those are very hard and,
Ludicrously expensive.
Ludicrously expensive, and they're not even working yet. And in some cases, they're just napkin sketches or, hopes and dreams in our
a work in progress. Yeah.
Yeah. Right. And so there's a spectrum there, but it's no longer is it like something that is many years and, lots of expertise for, I don't know if you agree, Leah, you might disagree, but that's
No,
what
I
think.
definitely agree. Yeah. There's everything. I mean, you could hook a assay up in yeast for minimal amounts of money, and it only takes a couple of days. Whereas, our lab, our graduate student that Jug and I share right now is trying to port this stuff into induced pluripotent stem cells, and good Lord, is that student spending all of our money?
Wow. I mean, that sounds really interesting. Anything with induced pluripotent stem cells is so interesting. We've done a few episodes just about that. But yeah, I mean, as someone that only recently learned about this, do you think it's going to get to the point where people in genetics know of MAVEs, or is it going to be more of that nuanced, if you know status? Like, is it going to be, it's not going to become as big as CRISPR, I don't think, because CRISPR, reached the lay people and just, all that. But within our genetic nerd community, is that going to become something that you think people will know?
It's a good question. I'll say what I think, and then Leah should say what she thinks, because she might disagree. I think that most people in the genetics community who are like thinking about functional genetics anyway, and are at like the PhD level, probably know about this method. At least they've encountered it in some way, shape, or form. As you point out, a CRISPR is like an example of what I would consider to be a foundation method for all of genetics. And I don't, MAVEs are not that, right? They're a way of multiplexing a particular type of measurement. That's my answer. I don't know what you think, Leah.
I had interpreted her mention of geneticists as clinical geneticists. And I think they will, because clinical geneticists who are interpreting variants and they're seeing these VUS are kind of desperate for information.
Yeah. Especially in peds. Yeah.
Yeah. And so I think, any source of, lots of information is going to end up on folks' radars in that sense. But, one of the problems we can tell you about is that we've kind of had this little bit of a last mile problem where we're generating all of this data. I think when we had just started publishing this, I had worked in BRCA1 and, it's a gene people know about and people get tested for. And we published all of this data that it was really accurate. And we're like, why aren't people using it? And it turns out that, supplementary figure 2 or supplementary table 2 is not something that your run-of-the-mill clinician is going to go look up. And so I think a place that has surprised both me and Doug about our careers is that we now are running like software development teams who are building the tools to be able to serve this to that community in a way that they can read it, in the way that they can understand that data. And that has been a very big surprise to me, where I thought, well, I'm just going to go hang out in my lab and generate this data.
Be with your research lab people versus a little bit more of my world of actually being in patient-facing roles where you're looking at this data or you're reading through a report and it's a VUS and you're like, I'm going to go and look at ClinVar myself. Look at, there's certain clinicians that will do that and more power to them. But then it's like, you got to start learning all of this. But no, I like that you both took the question differently to have the different perspectives there. But this has just been so fascinating. Obviously, I've just asked all these questions that I was just so curious about. But before we wrap, is there anything that you guys are remiss that I didn't ask you or anything you wanted to leave people with?
I guess I would say exactly where Leah left it is a really interesting place to consider, right? That is, clinical genetics is a really interesting intersection of research and getting stuff done in the real world, for lack of a better way of saying it. And like what I learned doing this work is that oftentimes if you have an idea that you think is a good idea, at least with respect to, research, making it matter in the world is like a separate step that requires very different skills and also just as much work as kind of giving the idea to the world and saying like, isn't this idea wonderful? It's going to make things so much better. Everyone might agree and say, like, it's going to make things so much better. That's great. Why don't you go spend the next decade of your life like doing that? And if you say no, then the thing might not happen, or it might happen a lot later. So that's kind of my takeaway.
Yeah, amazing. Well, thank you both so much. This has just been so fascinating. And now this is on my radar to see like, how this progresses. And again, just like, where we get to with VUSs. I think that's kind of been an ongoing conversation, and it will continue to be in terms of how long will we have VUSs, be a prominent issue. And yeah, hopefully by 2030-ish, we'll put a little ish on there for you, Doug. We won't be as focused on it in a good way, but I'm sure we'll have to revisit the conversation of just, how it goes. And as we mentioned, a lot of the resources we've mentioned throughout the episode, the symposium in Barcelona, ClinVar, so many different links we'll put in the show notes. But Leah and Doug, thank you so much. This has just been so fascinating.
Thank you. You've asked some great questions.
Yeah, thanks. It was a lot of fun.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.